to cover the second part of chapter 14 that discusses the regulation of the citric acid cycle. So we ended last time by noting if we look at the delta G values, not the delta G zero prime values, but the delta G values, we see that the citrate synthase, isocitrate dehydrogenase, and the alpha-ketoglutarate dehydrogenase enzymes have large negative values of delta G. So the artist or the author here doesn't give us the information, but the other values have delta G's close to zero, which means those reactions can be reversed. But these three reactions that I have my arrows pointing to have negative delta G values, so that's where we will regulate the pathway. We are, though, going to start before that into the entry into the cycle. So remember that pyruvate is converted to acetyl-CoA and carbon dioxide through the action of this large pyruvate dehydrogenase complex. So we have to consider the regulation of the pyruvate dehydrogenase complex as well as the other three enzymes in the pathway. So let's look at that. So here's the reaction. So pyruvate plus NAD plus coenzyme A will give us acetyl-CoA, NADH, and CO2. So it's an oxidative decarboxylation. And we have these three component enzymes of the large pyruvate dehydrogenase complex, which is shown in the upper left-hand corner. So we might be able to guess that that the NADH and acetyl-CoA would be involved in product inhibition. So acetyl-CoA and NADH will stabilize the T form of the enzyme. So in the T form, the pyruvate dehydrogenase complex has a low affinity for the substrate. So this is an example of product inhibition. And we also might be able to guess that we have stimulation of the pyruvate dehydrogenase complex in which the reactants, coenzyme A and NAD+, will stimulate the enzyme. And in this case, the coenzyme A and the NAD+ stabilize the R form, or the high affinity form of the pyruvate dehydrogenase complex. So there's the allosteric regulation that I just showed you, but the pyruvate dehydrogenase complex is also regulated by covalent modification or phosphorylation. So in this case, phosphorylation is an off switch. So there is a kinase, remember kinases, they catalyze phosphorylation reactions using ATP as a phosphate donor. So if we phosphorylate the complex, then the complex is inactive. A phosphatase, a specific phosphatase, a PDH phosphatase, will dephosphorylate the complex, and that is an on switch because removing the phosphate will activate the pyruvate dehydrogenase complex. So if we go back to our original reaction, so I've just sort of put a big circle around the pyruvate dehydrogenase complex to indicate that it's phosphorylated, and I colored it in red to show that this is an inactivation. So if you look in the inset in the upper left corner, you can see that we have the kinase, which accomplishes the phosphorylation. It's actually E1 that gets phosphorylated. And if you notice, the NADH, so the NADH and the acetyl-CoA that served to allosterically inhibit the NADH, the same things, the NADH and the acetyl-CoA also stimulate the kinase because the kinase is going to phosphorylate and inactivate the enzyme. So the enzyme is inactivated both by allosteric inhibition of the products, the NADH and the acetyl-CoA, and the NADH and the acetyl-CoA acting as allosteric stimulators of the kinase that will phosphorylate and inactivate the enzyme. So these things work together. The pyruvate dehydrogenase phosphatase will catalyze the hydrolysis of that phosphate. So I drew a green circle here to indicate that the enzyme is activated. So that's shown over here. So if we have the phosphate on the E1 and we have the specific phosphatase that removes it, then the pyruvate dehydrogenase is active, and that's shown in green. So here again, the NAD+ and the coenzyme A that we saw that activate the pyruvate dehydrogenase complex, they also, along with ADP and pyruvate, they inhibit the kinase. So the kinase, remember, would phosphorylate and inactivate the enzyme. So these reactants are able to stimulate the activity in the kinase and inhibit the kinase that would phosphorylate and inactivate the pyruvate dehydrogenase complex. So in that way, the NAG plus and the coenzyme A are working together, not just to stabilize the R form or the high affinity form of the enzyme, but to inhibit the kinase that would phosphorylate and inactivate the enzyme. So the hormonal regulation of PDH is currently not well understood, but these are some facts that I was able to uncover during some of my research. So in the fed state, adipocytes, the insulin appears to activate the PDH phosphatase. So that, remember, that's going to activate the pyruvate dehydrogenase complex. So again, the enzyme converts pyruvate to acetyl-CoA. We'll find out in a later chapter that acetyl-CoA is the starting material for lipids. So it makes sense that if you're in the fed state that you'd want to be synthesizing lipids. In the muscle cells, insulin activates the PDH phosphatase by a different mechanism. And the purpose there is different. The acetyl-CoA can enter the citric acid cycle to produce ATP for the working muscle. It's a different type of activation. Glucagon in the fasted state might inhibit the phosphatase, which would decrease the conversion of pyruvate to acetyl-CoA. All right, so let's go on and look at this reaction in a little bit more detail. So we have seen that if we carry out, so I have everything in my color right here, we can carry out glycolysis in the forward direction here and gluconeogenesis in the reverse direction. So we found that we could take glucose and we could oxidize it to pyruvate, or we could take pyruvate and we could carry out gluconeogenesis to convert it to glucose. So in some way, at least, we're reversing this pathway. Now, we had to use different enzymes to do that, and we needed more ATP as an energy source to do that. But we could go from glucose to pyruvate and pyruvate to glucose. Here is an example where we cannot reverse the reaction, even not with a different enzyme. So when we go from pyruvate to acetyl-CoA, I drew a big arrow right here to indicate that's the only direction that reaction can go, at least in mammals. I'll show you at the end an example where plants and microbes can sort of bypass this, but mammals can't. So we can go from pyruvate to acetyl-CoA. That's the pyruvate dehydrogenase reaction, and there isn't any other reaction or any other enzyme or any other ATP or anything that can reverse that reaction. So let's be very, very clear about this. So if you look here, you can see pyruvate can be converted to OAA using the pyruvate carboxylase reaction, and then PepCK converts it to phosphoenolpyruvate to convert this to glucose. But acetyl-CoA can't do that. There isn't any way to get from acetyl-CoA to OAA. There isn't any way to do that. And acetyl-CoA can't go backwards to form pyruvate. So this is the source of the information I gave you a while back, and that you need, if you want to synthesize glucose, you need 3 carbons. 2 carbons won't work, and acetyl-CoA has 2 carbons. So there isn't any possible way to get from acetyl-CoA to glucose, at least if you're a mammal. Okay, so let's look at the regulatory strategies because we want to be able to reason them out and not memorize them. So NADH and NAD plus balance is going to be important. If you think about the citric acid cycle, it's an NADH producing pathway. We will find out in the next chapter that NADH will funnel into electron transport, and then oxygen will be consumed. Oxygen is the final electron acceptor in the electron transport pathway. That occurs with concomitant production of ATP in oxidative phosphorylation. So that's going to come down to the demand for ATP. So it makes sense that if we have if we move down here, that ATP concentration is high when the energy status of the cell is high. So it makes sense that ATP will be allosterically, will act as an allosteric inhibitor of some of these enzymes. Whereas ADP concentration is high when the energy status of the cell is low. That indicates ADP is needed, and ADP can act as an allosteric activator of some of these enzymes. We're going to add another layer of regulation that we haven't talked about before. In many cells, a high concentration of calcium indicates that cellular energy is needed, so we won't go over the reasons for that now. But we'll just say that high calcium concentration is an indication that cellular energy is needed. So in that way, calcium stimulates some of the enzymes in the citric acid cycle. So the way we regulate the citric acid cycle is we control flux or flow through the pathway by three simple mechanisms. One, do we have substrate available? So it is a cycle, but we start with the first reaction with acetyl-CoA and OAA. So we have to have both of those reactants in order to start the whole cycle. NADH, remember we produce 3 NADHs per turn of the cycle, so that's considered to be a product of this cycle. So NADH can act as product inhibition. ATP could be considered a downstream product as those NADHs go through electron transport and oxidative phosphorylation to make ATP. And of course, we do have one substrate level phosphorylation step in the pathway that produces GTP, which is energetically equivalent to ATP. So NADH and ATP will serve as product inhibitors. And we also have some competitive feedback inhibition by intermediates further along the cycle. So let's get started. Okay, so we just talked about the regulation of pyruvate dehydrogenase, both allosterically and through phosphorylation. So let's look at that first reaction, citrate synthase. So just as a reminder, OAA plus acetyl-CoA are acted upon by citrate synthase, which catalyzes the reaction to form citrate. So this reaction works on substrate availability. Like I said before, you need OAA and you need acetyl-CoA. And ADP sort of acts as a substrate, not directly, but again, it's an indication of cellular energy status. So if you're using up a lot of ATP, ADP will indicate that energy is needed. So product inhibition, so a citrate is a product of this particular reaction. ATP and NADH, again, can be considered to be products further along the way. They serve as inhibitors of this enzyme. Succinyl-CoA is also an inhibitor of citrate synthase, and that's one of the metabolites further along the cycle. We should also note that citrate inhibits PFK1. So PFK1 is the major rate controlling enzyme in glycolysis, and glycolysis makes the pyruvate, and pyruvate is converted to acetyl-CoA. So we can look at glycolysis as being the pathway that provides pyruvate and then acetyl-CoA for the citric acid cycle. So here's another point of regulation that, that goes back to a previous pathway. What about isocitrate dehydrogenase? So here again is a reminder of this reaction. It's an oxidative decarboxylation where we lose this carboxylate group right here that's shaded in blue, and CO2 is the product. So we go from a 6-carbon compound to a 5-carbon compound. So that's alpha-ketoglutarate, and it's an oxidation. Again, we can look at this carbon right here, 1 bond to hydrogen, 1 bond to oxygen, and now we've got 2 bonds to oxygen. And here, those pink hydrogens and their hydrogens and the electrons will be donated to NAD plus to form NADH. So again, we have calcium and ADP acting as allosteric activators of this enzyme. And both calcium and ADP are indicators that the cellular energy status of the cell is low and that energy is needed. NADH and ATP. So NADH is an actual direct product of this reaction. ATP is an eventual product, so they both as product inhibitors of this enzyme. Next enzyme, alpha-ketoglutarate dehydrogenase. Again, another oxidative decarboxylation where we lose this carboxylate group right here, and the carboxylate group is lost as CO2, and the other product here is NADH. And similar kind of strategy. Calcium is an allosteric activator. Again, calcium is an indicator that cellular energy status is low and energy is needed, whereas succinyl-CoA and NADH, which are the direct products of this enzyme, act as inhibitors. So I'm going to show you this animated figure. Okay, so we start here. So this shows the regulatory points at the 4 steps where I showed you, the pyruvate dehydrogenase reaction, and then the 3 enzymes in the citric acid cycle itself. So the green circles represent sites of activation by calcium ions, and then the red octagons represent sites of inhibition by NADH. So NADH is in the circle right here, indicating that it's an eventual product. So if we look at the pyruvate dehydrogenase complex, we can see that that's activated by calcium ions, but it's inhibited by its products NADH and acetyl-CoA. If we move to the citric acid cycle itself, so the second site of regulation is the first step, where OAA and acetyl-CoA condense to form citrate, and that is inhibited competitively by succinyl-CoA and by citrate. And it's inhibited by NADH, which is not a product, but it is an eventual product of the cycle. Okay, moving on, the isocitrate dehydrogenase. So here, NADH, it acts as an inhibitor, and ATP also acts as an allosteric inhibitor. And calcium allosterically activates this enzyme again. So here, shown in blue, calcium and ADP, and they're shown in green. So the inhibitors are shown in red and the activators in green. Calcium and ADP stimulate the enzyme. Again, those 2 substances indicate that the cellular energy status of the cell is low and that energy is needed. And then the 4th site of regulation, the alpha-ketoglutarate dehydrogenase. So we have NADH and succinyl-CoA act as inhibitors, and calcium is an allosteric activator. Okay, so let's go back to the PowerPoint. So we can really look at this kind of strategy. There's a lot of details here, but the regulation of the cycle really depends on the pyridine nucleotide NADH-NAD+ ratio. So again, if that ratio is high, then we're going to slow down the pathway. But if the ratio is low, that means you don't have a lot of NADH and you have a lot of NAD+, and that means energy is needed. And the ATP-ADP ratio, again, if that ratio is high, we have a lot of ATP and we don't need any more, but if that ratio is low, we have low ATP and high ADP and calcium. So if we just look at those 3 regulatory strategies, that's going to help us understand how this pathway is regulated. So in a lot of the pathways that we've talked about, we talk about how is this pathway catabolic or anabolic. The citric acid cycle is what we call amphibolic, amphi being both. So it is actually both depending on how you use it. So if acetyl-CoA is completely oxidized to CO2, that's definitely acting in a catabolic way. But the cycle is anabolic because you can use intermediates from the citric acid cycle to make other compounds. So in that way, you regulate the pathway based on what is your need for those intermediates, for those biosynthetic precursors. So these are the anabolic reactions, and they're shown in red arrows. So we can use succinyl-CoA to make porphyrins, which is a precursor for making heme, acetyl-CoA, we'll find that out in the next chapter, is used to make fatty acids and cholesterol. When we get to the amino acid chapter, we'll find that we can make some conversions very easily between alpha-ketoglutarate and certain amino acids and between OAA and certain amino acids. So we can siphon off some of the intermediates that way. So again, I want to make sure, because this is also shown here, that if you can get to pyruvate, you can get to OAA. And if you can get to OAA, you can get to malate, and then you can get to glucose. But that, again, that means that you need a metabolite that's got 3 carbons or more. So acetyl-CoA will not do it for you. So if you are taking intermediates from the citric acid cycle, you need to put them back, because the citric acid cycle is the major source of free energy in organisms that oxidize glucose aerobically. So if you're constantly siphoning away intermediates, then you'll interrupt the cycle, and that won't help the organism obtain the free energy that they need from the oxidation of glucose. So if you take something out, you got to put it back. So reactions that produce any of the citric acid cycle intermediates are called anaplerotic reactions. So that just means to fill up. So basically, if you take things out of the cycle, you got to fill it back up. So those intermediates are not themselves oxidized. Remember, it's the acetyl-CoA that's oxidized, but their function is to boost the catalytic activity of the cycle. So let us go to another animated figure right here. All right. So here is an outline of our cycle. And here, this is what I showed you in the last slide about how we can siphon off citrate to make fatty acids or cholesterol, or alpha-ketoglutarate to make amino acids, or succinyl-CoA to make porphyrins. So in red, those are the reactions that are siphoning intermediates out, and the green arrows means we're putting things back. So we can take amino acids, convert them to alpha-ketoglutarate. The branched chain amino acids, isoleucine, methionine, and valine, their metabolism produces succinyl-CoA. Add chain fatty acids also produce succinyl-CoA. So we'll talk about that in later chapters. Aspartate, phenylalanine, and tyrosine, their breakdown produces fumarate. And there are certain amino acids that can produce OAA. So when you think about the anaplerotic reactions, making sure that you have OAA is going to be really important. Okay, so that's the end of the animation. So let's go back to the PowerPoint. So the PowerPoint actually, sorry, that animation didn't show this reaction right here. There's one anaplerotic reaction that's more important than any of the other ones that are shown here, and that's this one, where pyruvate and CO2, this is the pyruvate carboxylase reaction, produce OAA. That's the most important because OAA is needed to condense with acetyl-CoA in the first step. So it's really important to replenish OAA so that first step can happen, so that the rest of the steps can happen. So that's the pyruvate carboxylase reaction. So let's look at that in more detail in the next slide. Oh, just a minute. Okay, so I have boxed in green here my my other reactions that produce other intermediates for the cycle. Okay, so let's look at the pyruvate carboxylase reaction. So we have CO2 plus pyruvate. That requires ATP, and we get oxaloacetate. And the enzyme is pyruvate carboxylase. Remember, whenever you have CO2 as a reactant, you need the carboxylase reactions need biotin, and biotin is a cofactor that carries the CO2. And hopefully you remember that this was also presented as the first step of gluconeogenesis. So we're just going to borrow this reaction from gluconeogenesis in order to produce OAA. Now you know that means that not all cells produce or express, pyruvate carboxylase. So this is definitely a reaction that's happening in the liver. So the cycle does need both OAA and acetyl-CoA. So notice that glucose can give us both of what we need. So if we have glucose to pyruvate, and that's glycolysis, we can use the pyruvate dehydrogenase complex to convert pyruvate to acetyl-CoA. But we can use the pyruvate carboxylase reaction to give us OAA. So as long as there's a good source of pyruvate from normal carbohydrate metabolism, then the citric acid cycle can operate normally. Um, so again, both OAA and acetyl-CoA are needed. It does look like there's a one-to-one stoichiometry, but you know that you can get away with less OAA because OAA is regenerated. So you don't need 1 mole of OAA per 1 mole of acetyl-CoA, but you have to have some. Again, you don't need as much because the OAA is regenerated. But if acetyl-CoA concentration does increase, which it could if you were breaking down lipids, so we'll find out in chapter 17 that lipid is um, um, degraded to acetyl-CoA. So if we have lots and lots of acetyl-CoA, then the acetyl-CoA can act as a stimulator of pyruvate carboxylase to make sure that there's enough OAA to process all of the acetyl-CoA that's entering the citric acid cycle. So during exercise, this is a little bit different. We have glycolysis to pyruvate, and pyruvate dehydrogenase gives us acetyl-CoA. So here we have amino acids, and this could happen from protein breakdown. The transaminase reactions can convert pyruvate to and glutamate to alanine and alpha-ketoglutarate. So that's a way that you can boost the activity of the citric acid cycle in the muscle to fuel to produce ATP to fuel muscle contraction. So I'm going to switch now to talk about something called the glyoxylate cycle. So here acetyl-CoA is used to synthesize OAA. So I really want to emphasize that this happens in plants and microbes. Mammals can't do this. We don't have the glyoxylate cycle, we don't have the organelle, and we don't have the enzymes. So as I showed you before, acetyl-CoA is a breakdown product of lipids. And OAA can be used to synthesize glucose. But again, that occurs only in organisms that have the glyoxylate cycle. They are the only ones that can use lipid to synthesize glucose. Mammals cannot use lipid to synthesize glucose. That's not something we can do. So here is the glyoxylate cycle. So hopefully a lot of this looks familiar. So if we start up here with acetyl-CoA condensing with OAA to form citrate, and then citrate is isomerized to isocitrate, then remember in the citric acid cycle, we first lost 1 and then 2 carbons of CO2, but we're going to skip those steps. So there's an enzyme called isocitrate lyase, and we're going to break that bond right here, and we'll produce something called glyoxylate. And then the remaining molecule forms succinate, and then that can continue to form fumarate, malate, and then OAA. So if we have glyoxylate, then if we bring in a second molecule of acetyl-CoA and we have this enzyme malate synthase, we can synthesize malate, and then we have OAA, and then OAA can enter gluconeogenesis. So what we've done here is we've taken 2 molecules of acetyl-CoA and combined those 2 carbons to form OAA. So we've done that instead of oxidizing the carbons to CO2, which is what you do in the citric acid cycle. So again, these enzymes that are in green, they have the little green boxes around them. These enzymes are unique to the glyoxylate cycle. So if you don't have those enzymes, and mammals don't, then you cannot carry out these reactions. So here's another picture from a different textbook. So what it does is it shows the glyoxylate cycle summary, and it sort of grays out over here the reactions of the citric acid cycle that would take those two carbons and oxidize them. So again, if we start with acetyl-CoA condensing with OAA to form citrate, and then citrate being converted to isocitrate, we're sort of taking a detour and we're skipping those grayed out reactions in which the carbons are oxidized. And instead, we get glyoxylate, and then we bring in a second mole of acetyl-CoA to form malate, and right here the succinate can form malate, fumarate, sorry, and then malate to continue the cycle. So the net reaction here is that two acetyl-CoAs form one OAA, and then the OAA can be used to form glucose. So the acetyl-CoA arises from the breakdown of lipid. So this shows how breakdown products of lipid can be used to make OAA and can be used to form glucose. So please, again, only happens if you have the glyoxylate cycle. So here's the first acetyl-CoA. There's the second acetyl-CoA. So those two carbons combine to form OAA. And if we get to OAA, we can make glucose. So here's an example of how this would work out in a seed. So I study nutrition, so I'm interested in seed oils that form part of our diet. So what the food manufacturers do is we just take the seeds, and then you can press the seeds to obtain their oil. But if the seed is used to be planted in the ground to generate a plant, the seed stores oil because that's a really concentrated form of energy. So if we start here, so the seed oil would be broken down into acetyl-CoA, and then this occurs in an organelle called the glyoxysome. So again, we show acetyl-CoA condensing with OAA to form citrate, and then the citrate forms isocitrate, and then we break this bond right here to form glyoxylate, where in this picture it is shown in red. And then what happens is the succinate leaves the glyoxysome and then is transported to the mitochondrion, and then hops into the citric acid cycle. So here we complete the cycle here where a second molecule of acetyl-CoA reacts with glyoxylate to form malate and then OAA in order to continue the cycle. But what's happening over here is that since the succinate is forming fumarate and malate, then the malate gets transported out of the cytosol, and then malate forms OAA, and then OAA enters gluconeogenesis. So in the plant, that helps generate glucose until the photosynthesizing apparatus in green leaves can take over the process and generate glucose from CO2. So that means if you're an organism that is able to do both, then you have to regulate this by partitioning the isocitrate between the glyoxylate cycle and the citric acid cycle. So if I'm up here, here's my acetyl-CoA, again, condensing with OAA to form citrate and then citrate to isocitrate. So if isocitrate participates in the glyoxylate cycle, then you'll generate succinate and glyoxylate. And then over here, the isocitrate dehydrogenase will produce alpha-ketoglutarate in the citric acid cycle. So notice how you get very different products. So if you're if you form alpha-ketoglutarate, you'll eventually go through the citric acid cycle, produce NADH and FADH2, and then go through electron transport and oxidative phosphorylation to get ATP. But over here, you're producing OAA that can go into gluconeogenesis to form glucose, and then from glucose you can get to amino acids and nucleotides. Of course, you need a nitrogen source in order to do that. So the plant cell then has to partition this isocitrate between these two pathways. So when isocitrate dehydrogenase is inactivated by phosphorylation, then if the isocitrate dehydrogenase is inactivated, then what happens is the isocitrate is directed over to the glyoxylate cycle. But if you dephosphorylated, then the isocitrate enters the citric acid cycle. So of course you have kinases that carry out the phosphorylation and phosphatases that carry out the dephosphorylation. So you have to regulate the kinases and the phosphatases. So again, those enzymes are going to be regulated by the types of intermediates that indicate that energy is needed and we need the citric acid cycle to be operated, or are we under different situations where we need to synthesize glucose, in which case the lyase should be active. So I hope you enjoyed this discussion of the regulation of the citric acid cycle, and I will see you next time. Here are my references, and I will see you later.